This lecture is dedicated to the molecular underpinnings of biofilm infection. It's clear that biofilm infects human hosts in a much different way than the planktonic bacteria that we've learned. So what we want to do is go through the paradigm shift that scientists have had in evaluating how bacteria cause human infections. The first thing we know about chronic infections is there's excessive neutrophils. The neutrophils congregate in the area of the chronic infection, and they're dysfunctional. They do not fulfill their immune function. And the question is, why does that happen? Well, as you can see in this slide, there's host tissue where the arrows are showing excessive neutrophils, but then right above that is a very dense region of nuclei of very small cells, much more in the order of bacteria. That is indeed biofilm. And what we're going to try to do is link the biofilm attachment on the surface of the host, working through a number of different molecular mechanisms and not only causing dysfunction, but actually commandeering host immunity to fulfill its needs for nutrition, mainly through hyperinflammation. The first thing we see when we look at biofilm in the context of a host is that things are quite different. This is a illustration that was put out by Costerton and Stewart way back in 1999. And what they're trying to show is that once a planktonic bacteria attaches, it quickly changes its gene expression to be something phenotypically quite different. It changes from a white or a planktonic cell to a black or a biofilm phenotype cell. 800 different proteins in Pseudomonas. This is like a caterpillar going to a butterfly. Fundamentally different types of bacteria, same genetic material, but phenotypically quite different. And the behavior of the biofilm is quite different. It forms the matrix around itself, the self-secreted matrix, but also it incorporates host proteins like fibrinogen. The other thing that happens is that this matrix sequesters the biofilm cells from the host immunity. White blood cells can't penetrate, and what we see here from Lamb's work way back in 1987 is antibodies can't penetrate into the biofilm. Up in the from about the 9 o'clock to 3 o'clock position, the top rim of this, that's an antibody stain showing that the antibodies for Pseudomonas are outside this accumulation of Pseudomonas in the alveolar space. So we see that antibodies don't penetrate, and that's well vetted, and we know that's true. The question is, why does that happen? The next thing we know is white blood cells don't penetrate. This is Jeff Leeds' work, and what he's showing is that activated leukocytes, he's worked with macrophages and other white blood cells. They nestle down into the biofilm, and then they fail to carry out their function of trying to clear and phagocytize bacteria. So the question is why do the white cells not fulfill their immunologic duties in a biofilm infection? It's almost like these things are being commandeered or taken over by the biofilm itself, and that's what we'll talk about, the molecular. There's no question that slow penetration or the matrix that's around the biofilm cells prevents a lot of antibiotic and biocide degradation of the biofilm community. Where the planktonic community is totally naked, the biofilm has significant protection from the matrix, but there are also molecular mechanisms to prevent a lot of the reactive oxygen species and other tools utilized by host immunity to degrade the biofilm. So it's not just the matrix that gives the biofilm its power and its ability to resist host immune mechanisms. So that's what we've got, biofilm infection. It was first put out by Costerton and Stewart in 1999, and what they're saying is that bacteria follows a fundamentally different method or pathway or strategy to infecting the host than single planktonic bacteria. And so we've known that for 10 years, yet our molecular scientists were really struggling with this in the early part of this millennia. So from about 2000 to 2005, 2006, we were fixed in a planktonic paradigm. We thought that bacteria would get into the host, would upregulate its virulence factors, would upregulate its proteases, kill host cells, digest those host cells, use those as nutrients, and then continue the infection. That's what we think of when we think of a bacterial infection. What Costerton and Stewart are proposing in this illustration is a more parasitic infection where the bacteria takes over a host space that's caused by trauma, or in this case, cystic fibrosis. It will create more space by doing collateral damage, but in the end, it maintains a small area in a tissue that it can reproduce for prolonged periods of time, not feeding itself through killing tissue and degrading tissue, but rather feeding itself through inflammation and allowing the plasma to percolate up through it. So here are the molecular underpinnings that say that, in fact, Costerton and Stewart were correct. Now, this is an interesting article. This was by Torres in 2007, and I think this was the dawning of, hey, bacteria are pursuing two fundamentally different strategies. What Torres and his group were studying was the Staphylococcus heme sensing system, the HSS. And what they were showing there was that once Staphylococcus aureus sensed heme, it knew it was in a host environment. So there are receptors that the host hemoglobin would stimulate, and very quickly, within minutes, a whole set of molecular pathways would be released that would put more adhesins or proteins on the surface of the Staph aureus that would allow it to attach to very specific host proteins like fibrinogen and other epitopes or handles on the host extracellular matrix. So what we're saying is as soon as staph knows it's inside a host environment, it puts out these receptors so it can attach very quickly. Once bacteria attach, that's the first committed step towards biofilm formation. What we also see is that virulence factors in Torres's terms were paradoxically or surprisingly downregulated. That surprised him. You could tell through the whole article that he's going, now, wait a second. We got these knockout Staph aureus, ones where we take away very important pathways, basically biofilm pathways, and when we knock out those pathways, we get an upregulation of virulence factors, and that's what's on the right-hand panel for the knockout HRT variant of Staph aureus. So if we take out the heme regulatory system for the bacteria, it actually acts more planktonically, it acts more individually, and it will increase virulence factors and try to kill host cells. Now let's go back to the wild type on the left-hand panel. Why in the world would it not use its virulence factors? Okay, if you'll read the quote, it comes straight from the article. It says, in all the coordinated activities of the HSS and the HRT systems, it allowed Staph aureus to sense an internal host environment, resulting in the tempering or the decrease of virulence factors to avoid excessive host tissue damage. Now, why would a bacteria want to avoid tissue damage? That's what it's going to eat. Unless it's going to take a biofilm strategy, attach, maintain the host tissue, and feed off plasma exudate that it causes by hyperinflammation. So what Torres and his group came up with in this molecular mechanism is that bacteria very quickly are trying to temper their virulence factors, and they're trying not to damage host tissue. In this particular model that he used for causing infection. So that's a very important first step, and you see that the molecular guys are starting to come up with a different thought process on how bacteria infect host tissue. Well, then there was, in that same year, Hartle, working with cystic fibrosis, noted something that was incredibly important, okay, through a type 4 secretory system, and you see that on the left-hand part of the slide. Effector proteins and other effector molecules that are put out by the bacteria into the host environment would cause certain things to happen to neutrophils. So if you look at the right-hand panel, what we see is Pseudomonas aeruginosa is causing a infection, and what they're showing is it's outside the airway epithelium, maybe attached to the airway epithelium, but probably a biofilm phenotype. These bacteria secrete these molecules that call in neutrophils. The neutrophils come in because there's pathogen-associated molecular patterns, PAMPs. There's all sorts of things that stimulate toll-like receptor 2, and you get chemotactic factors that call the neutrophils in. Pseudomonas doesn't mind that if it's in a biofilm phenotype because, as we saw earlier, the white blood cells really can't penetrate biofilm. But what's interesting is that the secreted molecules by the type 4 system cause the neutrophils to do two things. Number one, it shuts down the reactive oxygen species mechanism, so they can't really attack the biofilm. Second thing is it knocks out the CXCR1 receptor, so chemotaxis goes away. Basically, the neutrophils could no longer migrate. But the final thing that Pseudomonas does is it fouls the receptors that macrophages use to clear the neutrophils. What this means is you have effete or dying neutrophils that can't be cleared by macrophages. What that means is when those, like in the top left-hand corner of the right panel, when the neutrophils are killed by the Pseudomonas, those dying neutrophils now aren't cleaned up by the macrophages, but they release elastase. That high elastase causes the CXCR1 receptors to degrade. Those fragments of CXCR1 receptors stimulate the toll-like receptor 2, which releases IL-8 and other chemotactic factors, and then more new non-affected neutrophils are called in, and that cycle it's in a positive feedback cycle, and there's no host regulation on that. So what we find through this mechanism, well described in Hartle's article, is that this is an important mechanism that biofilm uses to call in neutrophils and make these neutrophils non-functional. And it does it through secreted, well-documented effector molecules through a type 4 system. We've reviewed that in our article, Biofilms in Chronic Wound Inflammation, in Journal of Wound Care. So if you want a more complete analysis of excessive neutrophils and how some of these molecular mechanisms, we've got a lot more references in there. Finally, the type 3 system is the one that I think is so important for wound care doctors to understand. On the right-hand side, you see E. coli attached to a gut epithelial cell. And what that shows is that E. coli, through a type 3 secretion system, can insert molecules that basically take over ubiquitin pathways that cause this polymerization of the cytoskeleton inside the epithelial cells. And now there's a pedestal, an attachment that the host can't get rid of for the biofilm to attach to. Again, attachment is the first committed step of biofilm behavior. So now the E. coli has a firm footing on the epithelial cells. It injects through this type 3 system, and they're called T3SEs, so type 3 secretion effectors, and there's dozens and dozens of them, and they change not only between species, but Pseudomonas can choose to use some and not express others. So at the end of the day, here we have different types of bacteria attaching to host cells, and the first thing they do is they block apoptosis. Now, why would bacteria want to immortalize or prevent host cells from dying or shedding? Well, it's because they have to keep a firm attachment. Now, why is that important in wound care? Because one of the main things we see in our wounds is senescent host cells. These are cells that are alive, but they don't manufacture anything. They don't migrate. They don't mitose. They don't shed like they should. We don't have neutrophils lysing the wound bed and cleaning up the dead stuff like they should. So all of a sudden, it just like the light goes on. The bacteria must be usurping neutrophil function. The bacteria must be, through these molecular mechanisms, causing the host, the wound bed, to be senescent. It's not the host that cause those cells to do that. And here are multiple articles that talk about how, that clearly describe molecular mechanisms whereby these secreted effector proteins from type 3 systems go into these cells and through mainly ubiquitin pathways block, usurp, commandeer these cells to be senescent and give the bacteria a firm foothold that the host can't get rid of. This is a really important molecular mechanism, and it explains so much of what we see in chronic wounds. So that's the molecular underpinnings in a nutshell. The thing that brings it all together is this article by Kim in Host and Microbe, and it's worth reading. It's very, very enlightening. What he's saying is you know, he's mainly looking at Crohn's disease and ulcerative colitis, and what he's saying is Salmonella and Shigella, like in A in the right-hand panel, the A portion shows that Salmonella and Shigella, E. coli can all kill these cells. They can get into these cells, they can release virulence factors and kill these gut epithelial cells. But what he finds is in B is what bacteria mainly want to do. So there's that same E. coli, the same Salmonella, the same Shigella, and they make these pedestals and they attach to the cells, and then they secrete things that block apoptosis, or they immortalize these cells so they can't shed or they can't reproduce themselves. So they create senescent cells that they can stay attached to, and the biofilm community can function from, so it can sustain itself through inflammation and not through cell death. On the left-hand side are the multiple articles that he reviewed. And so the quote from Kim's article is very important, and that's what the takeaway message from this molecular talk that we're given today. And it says, in addition, bacterial pathogens use a variety of mechanisms that balance breaching. Now, breaching is planktonic. That's acute. That's killing cells. Okay, so it balances breaching the epithelial barrier with maintaining the epithelial barrier in order to promote bacterial colonization. So you've got the breaching planktonic behavior, and then you've got the maintaining or bacterial colonization or biofilm behavior. Two different strategies that bacterial pathogens use. So now here's the final statement. These complex strategies represent a new paradigm of bacterial pathogenesis. Well, it's really not new because Kosta and Stewart said it 10 years ago, but now we have the molecular that says, hey, there's two ways bacteria are behaving. One is to breach or to kill. The other one is to maintain and to colonize. And that is what we're talking about. That's planktonic versus biofilm behavior. And that's what we shared way back in 2007, that it appears that there's two different ways that bacteria infect. There's acute infections like the top panel that we all know, contamination, colonization, critically colonized, and then if it invades, it infects. We know that. and that's the way we think of infection, but it's much more complex in a chronic infection, and that's the bottom panel. In biofilm, the bacteria gets in, but then it quickly forms a microcolony. When enough bacteria is present, then there's signaling that allows it to take three-dimensional structure. It puts biofilm down around capillary structures and rises up off the surface. It incorporates host proteins and it becomes a sustainable functional colony in that host tissue niche. And in wounds, that's skin, and it can just seed itself through detachment fragments, or it can put out planktonic seeds back out into the environment, and that completes its life cycle. So planktonic, think of acute infection. Biofilm, think of chronic infection, knowing that there's no such thing as a pure biofilm or anything, but in essence, the molecular mechanisms we just talked about most closely correlates with biofilm infection. When would a clinician think of biofilm infection? Well, if there's a chronic wound, that's a given. But how about 2 or 3 days after a surgery when a wound starts to dehisce? Is that a biofilm infection, or does it have to go longer? Well, if you have any of these signs, like bacteria adhered to a surface, or if you can take some off and look at it under a microscope, well, that'd be the gold standard for diagnosing biofilm. But there's not enough money and time for a clinician to be looking at things with SEMs. So surrogate markers would be if it's resistant to antibiotics, or if it's resistant to silver and hydrophoroblue and iodosorb. So if it's resistant to biocides, well, then that would make one think that it's biofilm phenotype. And then another thing is if there's a large number of species in a small area, so you take a 5-millimeter punch biopsy and you have 8 different species of bacteria, and they're 10 to the 5th, 10 to the 6th concentration or numbers, well, then those can't coexist as planktonic, so you would think that that was a biofilm. And probably the thing that clinicians use the most, if it acts like a chronic infection, it's a biofilm infection. So if it waxes and wanes with exacerbation, responds incompletely to antibiotics, or if the tissue fails to heal, with secondary signs of it, the signs of infection are odor and exudate and a stalled wound. If it just fails to heal, then you think biofilm infection. Well, that's what we see in most chronic wounds, or they wouldn't be coming to our clinic. So it all comes down to bacteria can infect in 2 different ways, and if we take that one way, the biofilm way that it can infect, and put that into a category called medical biofilms, we see where it helps us understand what wounds are and what it means. If we take all chronic infections that have been clearly linked to biofilm, that's 17 million people that are affected every year. 500,000 people die from those infections every year. So we're talking about a really big problem. And wounds are a huge part of that. There are millions of people that get chronic wounds every year, and people do die from their wounds or from their treatment of their wounds, such as amputations. So what we find is that if we take this whole group of diseases and put them together and look at how clinicians over time have evolved their therapy, what we find is that we treat that our colleagues in different specialties treat their chronic infections very similar to the way we treat ours. It's with episodic doses of antibiotics, mainly empiric. They treat their chronic infections empirically, and then they treat them by trial and error. They'll try one treatment for 2-3 weeks, and then they'll try another treatment for 2 or 3 weeks. There is no diagnosis and treatment going on in medical biofilms. And that's mainly the limitations of culture. Current clinical cultures really can't diagnose medical biofilms. But at the end of the day, the way every specialty treats their chronic infection is by removing body parts. The sinus is stripped, the tonsils are removed, lung transplant and cystic fibrosis, the metal is taken out, the catheter is removed, the heart valve is replaced. You can see how that's going, and where we end up in chronic wounds is we cut limbs off. Now, the question I'd ask you is that what you want to do? If that's not what you want to do, if you don't want to remove a limb to cure the chronic infection we call a chronic wound, then what I'd suggest is understanding biofilm principles and then looking further into ways to diagnose and treat and manage biofilm so that it doesn't lead to that amputation. Thank you.